So today we're going to look at DNA replication and we're going to see how it makes copies of itself so that each cell can have the exact copy of DNA and have all the information for those particular organisms. So all DNA must be copied before cell division, which is mitosis, and we've went through the process of mitosis, so you know the steps of that. So as we go along, the DNA is copied and then it's divided, and then each cell has that exact copy of DNA in it. So each daughter cell will receive that exact copy, and this occurs during replication during the S phase of the cell cycle. And this is where the chromatin starts cooling into chromosomes, so it's replicated, it has its DNA strand, and it's going to go through the process. So this is the cell cycle, remember, that we've talked about. We have our G1 phase, which is in interphase, and this is where your cellular contents, excluding the chromosomes, are duplicated. Your S phase is where your DNA is duplicated, so you have 46 chromosomes that are all duplicated at this point. Then you have your G2 phase, which is where everything double checks, makes sure everything's duplicated correctly, and is getting ready for the process of mitosis. And then you have mitosis, which is your prophase, your metaphase, your anaphase, your telophase, and then we go through the final division, which is your cytokinesis. So there's three main steps to replication. The first step is where you have helicase, which is an enzyme. Remember, anything that ends in ASE is an enzyme. So helicase comes through and unzips the DNA strand by breaking those hydrogen bonds that we talked about earlier, and that is occurring between the nitrogen bases or the base pairs. So this creates two new template strands. Your second step is where you have DNA polymerase, again an enzyme that comes in and inserts new bases that complements the base that's already there. So if it's adenine, it would bond with a thymine. If it's cytosine, it would bond with a guanine. So it basically builds back your phosphorus and sugar backbone. And then you have step 3, which is DNA polymerase also. There's a polymerase 1 and there's a polymerase 3. And so this proofreads the sequence, and if there's any errors, it tries to find them and fix them. So all of your DNA strands we talked about before are opposite, which is anti-parallel basically. So one strand is always upside down in relationship to the other strand. So the two strands are anti-parallel to each other, and then that's how they break apart, and then the pieces are joined back together. So one strand forms in the same direction that the helicase unzipped it. The other one must go the other direction, and so it's going anti-parallel. And this causes fragments of DNA to form, which are then later sealed by a different enzyme. Sorry about that. Some of these videos are not working properly. Okay, so the result of your replication is that you have two new strands of DNA that are exactly the same as the original strand. So in this first picture, you have them together, then they separate, and then you have the nucleotides coming in and bonding the nitrogen bases together to form two new DNA strands. So each DNA strand now has an original copy of the DNA and the new copy or new strand of the DNA. So they have an old and new strand that make up the two different DNA strands now. So why is DNA called semi-conservative? And it's because that the DNA replication has an old part and a new part. And so semi means half or part, and conservation means preserving the old way. So therefore it's semi-conservative because it has an old part and a new part. So it's conserving half of the original DNA. So the DNA polymerase laying down new nucleotide makes one error every 1 million nucleotides. Sounds good, but there's 3 billion base pairs in each human cell. So that's a possibility of 3,000 errors each time our cells replicate. But the DNA polymerase is done, other enzymes come in and proofread it and try to correct those errors that they find in the strand. So after the error rate is about one error per 10 billion, or one error every 3 cell divisions. So your base pairing practice. For each complementary base below, give the correct complementary strand of DNA. and it's actually already done it for you here. So if you look, you can see the A's are bonded to the T's and the G's are bonded to the C's all the way down. So take a few minutes to look at this and get it in your head how they're bonding. So when you look at this picture, you can see that there is a helicase here on the left-hand side, which is a hexagonal shape in this case. And so that's unzipping the DNA. And then you have your DNA polymerase that's building your strands back together. And then you have your two new strands in the center part, and then the two old strands on the outside part. Okay, that is the end of our little practice.